A very good afternoon, everyone. To our faculty members, respected Director Sir Professor S.K. Singh, Dean Research Professor Ashok Kumar, senior teachers Professor T.M. Mohapatra, Professor A.K. Gulati, students, and all our online participants. On behalf of the Department of Microbiology, myself, Dr. Tuhina Banerjee, extend my greetings to one and all for this session. It just happened to come, I just happened to come over a news today reading amid COVID fourth wave scare, the black fungus is back. With speculations of a fungal pandemic in near future, the importance of fungal diseases, their diagnosis, can never be better emphasized. And therefore, in this context, we have amidst us Professor Anup Kumar Ghosh, a renowned researcher in the field of mycology from the prestigious institute PGIMER, Chandigarh. As a part of the tradition of this university, we begin any event paying homage to the great visionary, our founder, Bharat Ratna Pandit Madan Mohan Malviya. I will request our esteemed guest to offer flowers to the photograph of Mahamanaji. Without much ado, I will request Professor Pradyut Prakash, Head of the Department of Microbiology, to give the formal welcome address.
Good afternoon, all of you. On behalf of Department of Microbiology, I feel privileged in welcoming you all for attending this guest lecture by Professor A.K. Ghosh. I welcome our Honorable Director, Professor S.K. Singh and Dean Research, Professor Ashok Kumar on this occasion. I welcome all faculty members, residents, research scholars of IMS. I also welcome participants who are attending this event in online mode. We are thankful to Professor A.K. Ghosh for accepting our invitation for delivering an important lecture on utility of MALDI-TOF MS-based diagnostics in identification of fungi. For the last several decades, medical microbiologists are using conventional phenotypic tools for identifying bacterial and fungal pathogens from their primary growth, which took at least 24 hours, and in cases of fungi, it may take even up to a week to speciate any pathogens that's too partially. However, in recent years, matrix-assisted laser desorption ionization time-of-flight mass spectrometry, that is MALDI-TOF MS, has emerged as a potential tool for rapid identification and diagnosis of bacterial and fungal pathogens, which takes hardly 1 to 3 hours to speciate 40 pathogens. Recently, I came across a demo video uploaded by Professor A.K. Ghosh himself regarding the use of this instrument in diagnosis of fungi, in which his team has demonstrated in detail regarding the use of the instrument in identifying different fungi. In near future, the Department of Microbiology may get a MALDI-TOF MS-based instrument under IOE scheme with the kind support of our director and dean research. I hope you will all enjoy the extremely informative lecture by Professor Ghosh. Thank you.
Thank you, sir. May I now request Director Sir, Professor S.K. Singh, to welcome our guest with a bouquet. Thank you, sir. I will now request Professor Ragini Tilak, Department of Microbiology, to please introduce our speaker today.
A very good afternoon to all of you. It is my proud privilege to introduce Professor Anup Kumar Ghosh to this august gathering. He is professor in the Department of Medical Microbiology, PGI Chandigarh. He did his PhD in medical microbiology from PGI in the year 2000. He has been received many awards in his career. ASM Travel Grant Award in the year 2008, ICMR Travel Grant Award in the year 2013, Best Oral Post Paper Award in the Mycology category in Microcon 2019, Best Publication Award in the year 2016, 18, and subsequently. Apart from this, he has undertaken many projects as principal investigator and co-investigator. His area of research interests are use of multi-top MS in fungal diagnostic, epidemiology, rapid diagnosis, pathogenesis of fungal keratitis, genetic susceptibility of invasive fungal infection in human. Apart from that, he has published more than 75 articles in the various reputed journals. I now invite Professor Anup Kumar Ghosh to deliver his lecture.
It's okay. Good afternoon, you all. First, I would like to take an opportunity to thank you to Professor Ragini and Professor Pradut for inviting me, and I must be grateful to the director who is and the research academic dean who is kindly spending their time and attending. So today I'm going to talk about the use of multi-MS in fungal diagnostics. Actually, this is not a lecture. It's my experience, you can say. I was a postdoc in US, and with the Professor Chakraborty's request, I came 2011 and joined as a assistant professor in PGI. And the following 2 years after, in 2013, we got our multi-top MS by Bruker. And then I started my work, and by this time our group has published more than 15 papers in this area, and we have worked every area which is possible to use this Maditov for the diagnosis of fungal infection. And one thing I must ensure you, this is the era of technology, and phenotypic method we cannot rely, I mean, many more now, and we have to rely on this cutting edge technology, because this can, you know, reduce the time and labor. And I mean, there are so many advantages which I am going to discuss or share. Now, as the audience is not really a mycology audience, so I'll start from the very basics. Like till now, we know that there are estimated 1.5 million existing fungal pathogens, and out of that, 72,000 they are known, and 1700 fungi every year it is being catalogued, and out of these only 3 to 500 they are known for causing human pathogen. Now, this is very difficult to identify these fungi in the conventional method, because, you know, it is getting time consuming. Some of the fungi, and when we are identifying fungi, we are relying on their spore and hyphal, the arrangement. So in some of the cases, I'll show you, like in case of black molds, black fungi, they are not sporulating. So how you can identify? You have very limited, options. So you have to have the sequencer. It's also a high ending. But in all those cases, the MALDI can help, which I am showing. Now this fungi, it can exist in the three forms. One is yeast, another is molds, and another is the dimorphic. Yeasts, the name Candida, Cryptococcus, they are unicellular, sometimes budding. Molds, the Aspergillus, Fusarium, they have hyphae. And dimorphic, the example is Histoplasma capsulatum, Sporothrix. So it's very interesting organism. Like in the human body, when they infect, they are forming the yeast form, but in the environment, they are existing as a mycelium form. So there are three forms. And if you are talking about the infection, the fungus can infect superficial or cutaneous infection, subcutaneous infection, and systemic, or the then it is generally originating the fungi in the lungs and other organs. Now, this is a recent slide of the Gaffey, where you can see how the important the fungal infection is. If you look into this AIDS, if you look into this cancer, and then TB, asthma, everywhere you will see these are the fungal infections are, they are associated. And we also know that from the 10-20 years, previous years, we used to believe that, oh, fungal infection, it is only the immunocompromised patient, or some cancer patient, so they are only having the fungal infection. But now this is wrong. With the time, we are now start believing even an immunocompetent person, they can have the fungal infection. So we have to, make our knowledge current. What is the fungal infection, how it can infect, and how we can diagnose it. Now, what is the diagnostic method for the fungal infection? We can diagnose the conventional method as well as the molecular method. In the conventional method, direct microscopy and the histopathology and the culture. Now, when we are talking about the direct microscopy, the wet mount, like it is from the direct sample, or the slide culture means it is we grow them and we analyze, or we examine the spore and hyphae arrangement, and we can identify. Now, the direct microscopy, it is very helpful, like calcofluor staining. It can, identify, or it can, give your diagnosis within a minute or two. But there are certain problems, like calcofluor, you can get, false negative, in the diagnosis. And that Cryptococcus also we can detect with the direct staining. Histopathology, yes, this is another way we can diagnose the fungal infection. But in histopathology, you cannot reach up to the species level. So this is a drawback of the histopathology. The culture, yes, but it takes a lot of time. Like some of the fungi, it takes 1 week. Even black fungus, they take 3 to 4 weeks. So what would be your answer to the clinician? So you cannot wait in these days. Now, but there are certain media like the chrome agar, which has come into the market. It also takes 24 to 24 hours for the identification. But still, we are not relying that much. So now we know that the direct microscopy, it has a poor sensitivity. Recovery of the fungi from the cultures, it has also sports sensitivity, histologic diagnosis. It is difficult to get the deep specimens, and it cannot identify up to the species level. Now, the question is, why we need the fungal identification up to the species level? Now, I'm just showing another slide that what are the different, molecular diagnostic method. First is the nucleic acid based method. We know the PCR based detection, DNA probing technique, and DNA sequencing. So PCR based method, we can use the targeted gene sequencing and real time. So basically for the fungal infection, we are targeting the ITS region or 28S region for the fungal diagnosis. Now, in case of the DNA probing, we use the line probe, microtiter oil hybridization, microarray, and in situ. Now, another way of the fungal diagnosis is the biomarker based, or this is the basically the serological methods like the capsular antigen for Cryptococcus, cell wall antigen like 1,3 beta glucan, mannan, galactomannan, and the cytoplasmic antigen. But the last one, which is the topic of today, that is the proteomic based identification, or it is going to be done, or it is doing after using the MALDI of MS. Thus, it can be Bruker or Whiting. Now the question is why we need the identification. So recently, I'm sharing one of the recent experience from this institute. We just received 78 yeast isolates, mostly from blood isolates, and Dr. Agini requested that AFST and ID. So we, just completed this work, and you see, this is the Candida albicans. Out of 19, 6 they were resistant, and 6 they were resistant to fluconazole. Tropicalis out of 17, 3 resistant. Pelliculosa, utilis, Codemia omeri, they are showing resistance. Even Cruzii, they are intrinsically resistant against fluconazole, but we are getting the echinocandida resistance. Now the problem is the Pelliculus and utilis. Did you heard about this name? It is very, very new. And we are still now relying on the identification on the basis of phenotypic, means we are doing the sugar assimilation, we are doing the sugar utilization, we are doing the direct microscopy, and all. But these organisms, they don't have any direction. So how you can identify? So that's why she, send us the Candida species. And when we, use our MALDI, within a minute we got like Pelliculosa, we got the utilis. But the striking thing is, Pelliculosa, we used to know that they are very, very sensitive bug, but now they are becoming resistant, and it is very, very, shocking to us, because we didn't think about the Pelliculosa can be resistant. Now we have started, the collaborative with Dr. Agini, and we will find out what are the, reason for this resistance. So this is why we need the identification up to species level, and yes, MALDI can help you very much. So what is MALDI-TOF? This is matrix assisted laser desorption ionization time of light. Now, this can identify on the basis of ribosomal protein analysis. Now, the term MALDI was first, coined by, this Helen Kemp and Michael Carras, but it was actually the thought process or the concept of scientist Anhalt, who started or who using first time the mass spectroscopy for the identification of bacteria. Now, this is the two MALDI available in our country. That is, one is Bruker and another is Biomeru. But in my beginning of the talk, I'm just declaring I'm not advocating any of the machine, but as our experience with the Bruker, because we had the Bruker machine. So I am going to, present my data, all the Bruker data, because we did not have Biomeru, although right now we have Biomeru, we have both of the machines. Now, this mass spectroscopy or any machine, if you don't understand the principle, then you can't, really read the machine. So the principle of this machine is the separation of molecules based on the March to charge ratio, and it is very simple. It has 4 steps. Number 1 is the sample preparation. Means when you are talking about the ribosomal protein, means you have to burst the cell wall, or you have to, disrupt the cell wall, then whole the proteins come out, then you centrifuge and you spot it, and then you do the MALDI analysis. So first step is the sample preparation, and it is very, very important, because all the bacteria, it is not a problem, because their cell wall is very fragile. But if you talk about fungi, some of the fungi like black fungi or mold, even mycobacteria, their cell wall is very, very thick and rigid. So to break the cell wall, you have to think And then once it is solved, then there is not a problem. Then after you prepare your sample, you put it into your MALDI plate. Then this plate you have to keep it in the machine, and the machine charges the laser on that the spot. Then the ion generates, and this ion it runs through a time of flight, and in the end of the time of flight there is a detector which is capturing this the travel of these ions, and it will give you a spectra which is called the MALDI spectra. So this is the typical schematic. So this is the MALDI plate. First we have to spot, then machine the laser charge this, then there is an ionization, the ion this travel through the time of flight, there is a detector in the end, and finally you get a spectra. Now, what are the use of MALDI? So MALDI you can use the identification of bacteria, identification of microbacteria, identification of fungi, and typing of microorganism, antimicrobial susceptibility, and direct testing of clinical samples. So why I made this 4 red, because these all the areas we have worked and we have published. Now, the first step which I was telling you, this is very, very important. There are 3 to 4 types of sample preparation we till now have done. One is the direct cell smear, means you have the organism, you have the pure. Another thing is you have to have the very fresh and pure organism to identify. So just take the one, touch that colony, and you smear it on the MALDI plate, and then you put it the matrix, and you go and do the MALDI analysis. Another is the on-plate extraction. On-plate extraction means you smear it on the plate, then you put acetonitrile and formic acid on the plate only, and then after that you put the matrix and you analyze. And other extraction method is off-plate extraction method, means you are not extracting on plate, so off plate, so it can be mold or it can be yeast. So off-plate extraction method, what you do, just you take your isolate, the fresh culture in a microfiche tube, you wash this, then you dry it and add acetonitrile, formic acid, and we are adding the glass beads and vortex it vigorously to get it burst, the disruption of the cell wall. And then after some time, you centrifuge this, spot it, and then you do the MALDI analysis. Now, in the MALDI top, I told you that we have to uh, prepare the spectra. So this is a typical. This is a typical when we are doing that test. So here you spot it, and when you charge your machine with the laser, it is the spectra it is generating, and when the spectra generates, it always compare with the database that I will talk about, and then it will give you a score. Like what is the Bruker score? That is 2 and more. It is a species specific identification. 1.7 to 2, it is genus specific, and below 1.7, it is not reliable identification. So your score should be above 1.7 and above 2. So species level is 2. Now, this is the main thing, that database. So what is the database? What company did, they have collected the pure culture of ATCC or CBS isolates, and they extracted the protein, and they acquired the spectra, and they stored in a database form, and they added it to the machine database. So whenever we are getting any Bruker, we are also getting the database by default. So whenever we are trying to test our organism, the extracted protein, it gives us a spectra, and that spectra it will match with the database, and it will give you the score. Now the question is, if there is no database, then you will not get any score. So that is the another area we are working on, because the fungal infection, the variety of fungal infection in India, it is unbelievable, and we have created a huge database. It is better than Bruker, and we are willing to share it to the different institute in our in a country, but the condition is you have to have the Bruker system, because it is not compatible in the biomarker system. So this is a database. Now this is a schematic, that is you have the pure growth and put it into the plate, then you do the margin off and it will give you the score. So if you have a pure growth, within 15 to 20 minutes, maximum half an hour, you are getting the species level identification. That is the beauty of this machine. Okay, I know that this is a little expensive. So this is our PGI machine. So now you can also tell me, so this is the spectra of Candida albicans, this is the tropicalis, this is the cruziae and glabrata. So what you see, are they similar? They are all different. And we have seen species wise, they are very, very particular. This is Candida albicans from 4 different patients. They are all blood isolates. Is there any difference? There is no difference. So that is the beauty of this machine. So this is the spectra. We know this is perfectly fine species wise. Now, when we got this machine, we standardized the technique, and slowly we started publishing our data. So this is the first publication we have made, that is the matrix assisted laser desorption ionization time of flight mass spectroscopy for the rapid identification of this. In this work, initially we started with PCR confirmed yeast isolates, and then we re-identified by Mali. So you can see there are Candida rugosa, Candida auris, Candida Codemia omeri, Candida vishwanathiae, the green one, they were not in the database. So I'm talking about in the year of 2014. So immediately the Bruker system, they are open. If you want, you can make your database and you can put it in your machine, but separate way. It will not go and attach to the Bruker database, but it would be your in-house database. So we have immediately, incorporated in our in-house database. And in this study, we started with 354 yeast isolates, and then we standardize the technique, and then we randomly validate with the 367 isolates. But you can see the name of the organism: Candida utilis, Candida lipolytica, Candida caffeid, Candida humuliniae. So many, even we did not hear about all those organisms, and we did not thought about that this could be a human pathogen, this could be a blood isolate. But as we have MALDI now, and the database is expanded, and database is really, really big, so even the very, very rare Candida species we can easily detect. Now you can tell me, you can ask that still if it is not there, there is a possibility. So in this work, we saw that there is database unavailable, that is Wicarohemomyces anomala, which is Candida pichia, pelliculosa. Then another 2 organisms, they were not in the database, and we immediately put it into the database. But in this paper, what one striking thing we have done, because we we asked the Bruker that whatever the database you are using, it is not perfectly right. Even 1.75, 1.8, it is giving us species level identification, and we statistically proved that. That's what I wrote here, that even the log score is more than 1.7, we can easily identify the Candida species level efficiently. And the sensitivity and specificity of this test is close to 100%. Now this paper is widely cited, and till today we have got 120 citation. So this is just a humor slide. Previously, when we didn't have the MALDI, our technician is like this. Now our technician is a funny book. So in the morning they come, they put it the test in the MALDI, and by the lunchtime they get free. So but when we standardize this method, the striking thing is that time there was no Candida auris. Now most of you all know that Candida auris is a multi-drug resistant. People are talking about it, but that time we did not know. So Codemia omeri and the Candida vishwanathiae. So later on, we published like this is a M.R. Shivaprakash paper, Candida auris candidemia in Indian ICU analysis and the risk factor. So in that study, we have seen that out of 1400 candidemia cases, 74 or 5.3% they were Candida auris, and they were from 19 out of 27 ICU. Now, in that study, we found that isolates were resistant to fluconazole, that is 58%, amphotericin 13.5, and caspofungin 9.5. But now the resistance has gone up really, really high. Another study we published that Candida Vishnunathia, this is a very nice story and I can share with you. In the year of 13-14 and 14-15, you know, from the December to April, in this period, we were getting isolates from the blood, and our Maldi was, you know, identifying as Candida Vishnunathia. Although this particular organism was first identified from VP Chase, Delhi, and worldwide, 21 report available until that time we published. So when we talked to Professor Chakraborty, initially he said, no, it cannot be. So we did the sequencing and we got, this is Candida Vishnunathia. And they were all sensitive. But the next year, again, the same time, 2014 and 15, we got another 14 isolates from the blood, and they were fluconazole resistant. Means there was no cutoff, but they were showing high MIC against fluconazole. So that is the beauty of this Maldi machine, because if we didn't have Maldi, and this is very closely related with Candida tropicans. So initially we were thinking that it could be a Candida tropicans, but finally Maldi, and thankfully we have the sequencing, we got the Candida Vishnunathia. So if you have this machine, you won't miss this kind of, you know, incidences in your center. So when we standardize, we had our job, that is the identification of mold, because yeast, to work with yeast, it's very, very easy and we really manage. But this mold identification standardization, it took us years, and finally we standardized, and now we published this article also, and we are following this method in our routine lab. So in this method, just I tried to show here that we use the different protocol, but whatever our modified protocol, that worked better, and finally we got the good procedure for the identification of mold. In this method, in this donut pie chart, you can see we started with 153 isolates for the Maldi identification. Initially, we identified the 123, but when we checked that the below part, they were not in the database, we immediately updated the database, and finally, after updating the database, when we identified, we got the 100% identification. So this is the way we standardize the mold also. When we standardize the mold, then our immediately, the next challenge was the black mold, because black molds were more challenging because they grow, slower. There was no sporulation, and then we started working with the black molds, and we published this paper in the proteomics and clinical application, that is the Malditov based identification of melanized fungi in faster and reliable after the expansion of in-house database. So in this case, what I want to show you, when we started this work, we had only 8 isolates in the database, and you see the number. So database is always been enriched when the number of the organism in the database would be more and more. This many number of then the database would be richer. But here you can see number 1, because they did not have that isolate. So that's why. But when we started, and we with the DST, I have got a grant, and with that grant, this is showing here 26, but right now we have 31 different species mold we have in our database. Now here we have showed that even with the DNA sequencing, Malditov MS is faster for the identification of black mold. Here in this table, I tried to show that there are time required for the conventional method of black mold, but some of the organisms like Epicoctum niagara, then the Lecidoploidea, Theobomi, and others, they don't sporulate. So that's why you cannot identify them conventionally. But if you are trying to do the sequencing, it will take even more time and they are more expensive. Even here I tried to show that different organisms species wise, they have the spectrum of protein spectra is different. Now the MALDI-TOF, it can be used for the typing also. When you have the different organism, the MALDI-TOF, you can do the typing. Here this is the same paper of this black fungi. We have, when we MALDI based dendrogram we created, we found that all the 26 different species we have created the database, they are different and they don't mix each other. Now, later on, we have also expanded the database with the Malassezia species. The MALDI-TOF, they had the only database with the Farfar and then the Restricta, but we have added the different. Even in Malassezia, there is one new species, Malassezia odonolucii, which is the new species we have, first explained and published. So with the Professor Chakraborty's name, we named it Malassezia odonolucii. So now we have a really good database of Malassezia. Now the last, the before last, that is the antimicrobial susceptibility testing. Like now we know that antimicrobial resistance, it's a problem in case of bacteria and fungi. So as I am working in the fungus area, I have worked on the drug resistance detection, resistance isolates detection by using the MALDI-TOF. So we have published, like one of the publication, the rapid detection of fluconazole resistance in the Candida tropicalis by MALDI-TOF MS. So here we started with 15 fluconazole resistant and 19 sensitive isolates, and we found that if we challenge this organism 4 hours with the drug, you will see the changes in their protein spectra. So here we tried to show that in case of Candida sensitive, tropically sensitive and resistant, after challenging the drug, there is a spectral changes which the MALDI can detect, and we can easily detect the resistant isolates. So this is a different way we try to prove that our technique is better. And this is the MPCC, the correlation between MPCC and MIC, which is also good enough to prove that. Now, another paper we published, that is the stable isotope labeling and approach of MALDI-TOF MS-based rapid detection of the fluconazole resistant and Candida tropicalis. Now, this is a very interesting, this is a concept, a different concept, like the resistant and sensitive, always they are multiplying, when you are growing them. But if you use the, isotope, although they are not radioactive isolate and not very, effective or toxic for the human being, so you can easily detect this resistant and sensitive isolates by using this technique. Now, the last, that is the direct identification from the clinical sample. So, you know that now, whenever you are doing the MALDI-based identification, you need a pure culture. But pure culture, you have to, isolate from the clinical sample. It will take some time. So, I mean, if you can cut down the time, it will help the clinician, and then that would be really help for the clinician. Now, why it's not moving? Now, when we started this direct identification, we started from the blood culture bottle. So the idea was, previously what we used to do, we get any bottle that is flagged, we take this 10 microliter of the blood sample from the bottle. We used to do the gram stain, and gram stain, it will clearly show whether it's a yeast or it's a bacteria. Then you go and you do the MALDI identification. But here, if you see the culture again, when we have the positive bottle or the flag bottle, we used to subculture, and it took again the overnight, so it was getting delayed. But what we have standardized it, that now whenever we are getting any flag bottle, we do the gram stain, and if the smear is coming yeast, we take out the 4 ml blood, we process it, and we do the MALDI based identification, and from the same pellet we are doing the AFST. So by that way we are cutting down the time from 72 hours to 24 hours. So this is the paper we have published, that is a identification of and broth microdilution antifungal susceptibility testing of yeasts directly from the automated blood culture. So in this paper we have identified the broth dilution and antifungal susceptibility. So we have identified the direct ID, and in this direct ID we use the lysis, means the 0.5% SDS, and from there we did the AFST, and we standardized with the 28 samples, and then we validated with the 70, and we are now in the routine lab. Now this is the schematic of that our publication. That is, initially when we get the yeast in the blood, then we make the pellet, and this pellet we are again cleaning up with a different procedure, and finally we put the AFST. So the next day we are getting the AFST result. So this is the way we can make the identification AFST faster, and we are now doing this routinely in our laboratory. Now this is a different organism we have identified directly from the blood culture bottle, and the direct AFST we got the 100% categorical agreement with culture AFST for posaconazole, anindole, fungin, for amphotericin B, voriconazole, and fluconazole. Now this is another slide. It's very interesting. I compared this conventional DNA based techniques and the MALDI and the price comparison. This is the conventional we are getting for candida species 2 to 5 days, DNA sequencing 2 to 3 days, but MALDI based, if you have pure culture, you take only 30 minutes. The price is also conventional 4 to 500, DNA sequencing almost 750 to 1000. But if you have a MALDI, it needs only 10 rupees, 5 to 10 rupees for one identification. So it is that cheaper, but it's an one-time investment. Now, the database, I told you that with the help of DST, we have created a huge database. So initially, we incorporated the isolates, like from the different centers, from the different source of samples. So in one organism, we included 10, one organism, 10 different organisms, and we created the database. So this is the existing Bruker database. That time I'm talking about 2-3 years back, and this is our database. So this actually Bruker, they are very interested to incorporate our database into there, but as we are working in a government institute, it's not that easy. I'm still waiting. Now I have told so many things good about the MALDI talk. Now there are some limitation also. First thing I told you, it's expensive. It's more than 1 crore. But if you can, invest one time, it will make your life so easy. Now database, it's a great limitation, and it is company specific. As I told, that you cannot use the blue card database in the bioMiro database. Now identification organism also from the direct sample, it was a challenge, but we are working on it. We have already solved the blood isolates. We are now working from the tissue sample, the organism. But one problem of the MALDI based identification is you need the certain number of organisms for the making the good spectra. So blood we are getting, but in case of other like tissue or pus, or even we have tried with the CSF, we are not getting that many organisms which can create that, spectra which through which we can identify. So it's challenging, but we're working on it. Now the last one is the mixed culture, because sometimes you can get the mixed infection. So what would be your answer? So that way, we are facing problems, but yet yes, there are problems. We are working with it, but as a whole, it's a really nice experience. So I'm just experiencing our, I'm just sharing our experience, how we are helping to the other centers. Like now, the JLN Medical College, Dr. Vijay Lata, she sent her PhD student with, trichosporin 80, and then she sent with the only species. And then that particular student, she came our center and worked for 1 or 2 weeks, and we got all the speciation done of the trichosporin, because the Bruker, they have a very good database of trichosporin. Now, Dr. Savitri Sharma, one day she called and she said, Anup, we have so many, black fungus which we isolated from the cornea, and they are just sitting here because they are not sporulating, we cannot identify. So I said, okay, send us. And we tried, and you see 56.2% out of all those we have identified. And those which were not in our database, after we sequence them, we added. So this is the way we are enriching. Even Dr. Ragini, I think 2-3 years back, she sent us more than 80 isolates, and within couple of days we have identified all. Then another, the medical college Haldwani, one of the student came and she worked on the dermatophytes. And this is also our experience also, because people are coming with different isolates. Sometimes we do not have those isolates. So we are identifying, we are adding it into our database. So that way, our database also getting richer and richer. But we are really, happy that we can help the people in this way. Now, the take-home messages are: the MALDI-TOF MS can be used in the routine microbiology lab for faster and reliable identification. Rare pathogens, which we have even, do not have any idea. If your database is available, even rarest pathogen also you can identify. This is a one-time investment, and it is very inexpensive if you have my machine in your, because it doesn't need any consumable. So what consumable you need? You need the acetonitride, you need the formic acid. It's a couple of thousand, that's it. And then it can be used for molecular typing, it can be used for the resistance detection, and then the direct ID from the blood sample, we can show that the turnaround time, it can be cut down from 72 hours to 24 hours, and the proper and use and timely report can help the clinician immensely. So if we can use this multi machine wisely, we can really help our clinician, because clinician, what they wants to know, they wants to know what antifungal we are going to use. They really, are not that interested whether it's a Candida pelliculus or, but yes, Candida auris, if it is Candida auris, now they are also interested, because Candida auris, everybody knows this is a multi-drug resistant. And if one drug is, resistant, then what we can use the other drug. So that way, microbiologists can help the clinicians this way. So this is our team. You know that Professor Chakraborty has been retired recently. Dr. Shiva, he is in charge now. Myself and Dr. Harsimran. And this is my PhD students. Saurabh Das is working on drug resistance mechanism in Candida auris. Yamini is working on fungal keratitis, the rapid diagnosis. Brinda is working on the biofilm and drug resistance in tropicalis. And Snigdha is working on heteroresistance and tolerance in Candida tropicalis. So this is it. Thank you very much for your attention. And I will be very happy if you have any questions which I can answer. Yes, yes.
Good afternoon, sir. Sir, you are doing it on the basis of protein.
Yes, yes.
So, do we need to purify the protein?
No, no, not at all.
Okay.
No, not at all.
Directly from
Directly,
the
you just disrupt the cell wall, whatever it is coming out, you centrifuge, and from the supernatant to spot it. There is no purification in it.
So, doesn't it have problem with the different lipids that are in the cell wall, the maltitol, or you need to isolate from the lipids?
Now, what's the question? Lipid, it will problem. But we are creating only the protein-based spectra.
Okay.
But the MALDI, now the recent MALDI talk of Bruker, they have got another module. Through the lipid profile also we can identify the microorganism. So that particular module we don't have, but possibly we are going to get it, and I'll start working on that. I'm very, very excited.
Nice presentation, sir.
Thank you.
Like from the blood sample, how are you identifying the fungus?
Pardon?
From the blood sample.
Okay, blood sample. So what happened in the blood culture, automated blood culture bottle, when this is a flagged, we take out that bottle, we take 10 to 20 microliter bottle after proper shaking, we do the gram stain, okay? And if the gram stain shows us that yeast organism is there, we draw 4 ml and we make the pellet, okay? Then we lyse the blood, because you have a lot of RBCs and all. Yes, but we are purifying that and we are making a pure pellet of yeast. That is the thing. You are right. There might be so many impurities which could really interfere, but there is a procedure we have developed through which all the impurities we can exclude, and we have the pure yeast pellet. 2 hours. 2 hours. Yes. There are 3-4 steps. It takes 2 hours. You will get it.
Sir, first of all, thank you for such a nice presentation of yours. I have one question which I would like to ask, that how do you troubleshoot in the situation when you get polymicrobial isolation, or you get a new spectra probably, but you are not sure whether it is a new organism or it may be because
That's
of some,
a good question.
mixed organism.
Yes.
So how do you troubleshoot those situations?
Okay. First, I'm going to answer your last question. Like when there is a new organism, how we will troubleshoot? We will do this, try it 2 to 3 times. At least 1 time, no result. 2 times, no result. But it looks that this is pure under the microscope. There is no problem. Then we immediately send it for the sequence. Sequencing, it will give you the proper identification. And then we go back and check what the organism is in our database that present. If it is not, immediately we incorporate it into the database. First thing. Second, your first question was polymicrobial. Yes, it's a problem. In our experience, we have seen when we are standardizing the direct blood sample, twice or thrice we get Candida glabrata and Candida albicans together. Okay. So what happened? It's very interesting. When we are this thing getting the score, first 3 Candida krusei and second glabrata, second 3 Candida albicans, and we don't know why, but we are getting. But yes, if you have this kind of situation, then we go for the and sticking it to the chrome agar plate, and we isolate. But it's very less. Although it is not, I mean, it is not, you cannot say it's not there, but it's a problem.
Hello, sir.
Definitely. Confirm your isolate. This should be pure, and whenever you are doing the MALDI identification, it should be fresh. We are telling the people not to keep it in the refrigerator to get a good identification of MALDI.
Hello, sir.
No, because we are using the protein, but virology, but people are using for parasitology these days. Even acanthamobi, they have the database, but virus, I don't know, they don't have. But MALDI is not very good, because I told you, for the MALDI identification, you need the organism with a certain number, and then you get the extracted protein, and then you are doing the identification. So virus, there is no database. So that's why you cannot, because it's not useful for the virus.
Yes, online attendees, you can please ask your questions.
Sure. Sure, sure.
Hello.
At least actually, in our center, we don't have that experience. I don't know about the, but another thing I'm telling you, the Thermo Fisher, they are these days marketing the septicizer, no? That the plates, the drug plates. So let's say you have a Maldi, but you don't have the, this thing, Thermo Fisher machine. Only thing, if you have those drug plates, you just inoculate, and you get the next day the FST. That's what, no. No, I told you that whenever we are making the database, it should be pure. I'll try. Yeah, that's a good point. Yeah, I didn't think about that checkerboard. That's a good. Yeah, 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 yeah. Yeah, this is not that cumbersome. Good point. Yeah. Thank you.
I think this is the power of newer diagnostics like multi-top, which gives us results in few hours time. And I come from clinical side, newborn babies. I deal with newborn babies. So in your slide, you showed the different group of patient population who are vulnerable to fungal infections. But I think newborns were missing from your slide. And in neonatology, we find especially babies below 1 kilogram who stay in NICU for a long time. By the end of 7 to 10 days, they suffer from fungal infections, mainly Candida, but it takes time.
Yeah.
Long time to get the report. Many a time we see the baby may die by the time the report comes. So I think this has a huge potential, multi-talk, in improving the patient care. Not only that, it will reduce mortality and unnecessary use of antibiotics.
Sure.
Yeah. It will also contribute to antibiotic stewardship. We can use antibiotics judiciously and less toxic and less expensive antibiotics. One disadvantage you said is high cost.
Yes.
But the running costs are much low. And there is no reason why in BHU we shouldn't have this facility. This is a high time, I think. Another disadvantage I find is you isolate organisms which are very difficult to pronounce. Those fungi.
The clinician for the clinician.
I think we need some simplification of taxonomy. So these become more understandable.
Sir, you don't understand the fungal
taxonomy.
It's a mess.
So that's what I want to say. Thank you very much.
Okay. Yes, I'm sharing one of our experience, because when we're working, you are having so many experiences. Like when we're standardizing the direct blood bottle. So one day in the evening, my student reported that, sir, today we got a Candida auris. So when we go back and found that patient, one 6 years old boy. So he was on fluconazole last 2 weeks, 1 week, and he was from Ludhiana, and his fever was not controlling, and they referred to PGI. So PGI, he came in the evening, and they sent the blood immediately in the blood culture bottle, direct Maldi. We got Oris. We called the clinician. They changed the treatment to echinocandin. Patient got okay within 2-3 days. So this is the advantages. Clinicians also, they are really looking to us that what we are giving the report. 1 crore 50 lakhs. Sir, these days, I mean, people are really buying, because in PGI AIMS we have. Private setup, Lal Path they have, and but sir, it's a nice machine. Definitely, sir. Not guideline. You see, please come to us. We will teach you. Yeah. Definitely, sir. Wherever they, even now, I am an expert for all the centers in the government setup. They are buying, no? Yeah. Sir, we are giving training to the people. We are giving. If anybody wants to come and learn from us, you are most welcome. PGI is open for everyone.
Any of our online participants? You can put it in the chat box, or we have unmuted all. You can unmute yourself and ask the question.
Sir, how you make the database for new species?
Okay, yeah, that's a good point. The database, what we are making, first we have to confirm 100% that the organism is such and such. So for the confirmation, always we go for the sequencing. So once we get confirmed, then we extract the protein and we spot 24 times, the same spot 24 times in the multiplane, and the machine acquires all the 24 spots, and out of 24 spots, 20 spots machine they only choose, and then they mix up and they make one spectra. That is the database. So that is all the software they are doing. Our job is to make sure that particular organism extract the protein, spot it. That is our job. The rest are the machine.
So we have one question like, can we detect microbes from skin scraping or nail scraping directly from antibiotic?
We haven't tried yet, but yes, definitely. But I told you that any organism which we are trying to detect by MALDI, it should have a number. So I'm not sure whether skin scraping, we are going to get that number. Sample amount is similar to conventional method means? Especially blood. Okay, actually, yeah, I know that what you are asking. I already answered this question, because for MALDI analysis, you need when we standardize the yeast, we need 1 million cell, like 1 × 10⁶ cell. So we use this for the extraction, and then from there we go. But when we use the blood culture, and that's what we standardize. We standardize from the 2 ml blood, 4 ml blood, 6 ml blood, and then we found that 4 ml blood, it is giving us consistent result. And then we count it, and we are getting with the 4 ml blood, we are getting that 1 million cell. So that is for sure. And what is the? Yes, ma'am. Our group, the antibiotics. What is the method for antibiotic sensitivity testing by Malita? Yes, and antibiotic sensitivity means for the bacteria it is different, For the fungus, it is different. Bacteria, we are detecting the MRSA resistant. So in case of bacteria, there is the drug is, it is giving the peak. So that way we can detect. But in case of fungus, we are challenging the organism with the drug, and when we challenge the organism, we are noticing or we are seeing there is a protein spectrum profile change, and that way we can detect the resistant isolates. Okay. But anyway, all the online this thing, if they can write me if they have any issue. I mean, if I know, I'll address them.
So I presume there are no more questions from our online attendees.
So for black fungus, what we do if we not getting in the Maldi?
Pardon? Pardon?
For black fungus, sir.
Uh-huh.
If we not getting the ID in the Maldi, then what you are doing?
Actually, black fungus, yes, we are getting. Black fungus, the problem was Bruker, they did not have a good database. But now we have created a good database, and you can identify by Maldi, the black fungus. Only thing you have to, vortex extensively, and then only you get the black fungus. Another issue I can discuss, in the black fungus, when you are doing the protein extraction or DNA extraction, as it is, producing the melanin, your DNA is black in color, your protein is black in color. So that color, it might interfere. In the PCR, black fungus PCR, I mean, is really, really not that high, because that particular color or melanin, it is interfering. In case of protein also, so we are diluting it, and the diluting it until it is going to give us the clear result. So that is one thing.
Thank you, sir.
Okay. Okay. अच्छा, ये खोल दीजिए.
Thank you, sir. We conclude today's session and would like to thank our speaker, Professor Anup Kumar Ghosh, for this highly informative talk, and also above all for accepting our invitation. I would also like to thank Director Sir and Dean Research Sir for their valuable time. This event would not have been possible without the efforts of our HOD, Professor Pradit Prakash, and Professor Ragini Tilak. So thank you, sir and ma'am. I also thank the staff, students of Department of Microbiology and the telemedicine team for their cooperation. And lastly, and most important, my sincere thanks to all the attendees present here and who have joined us online for making this event successful. Thank you.